Hey guys, welcome back. I hope you're enjoying the mini course so far. In this video, we're going to be talking all about Stage 2 biology. Biology is by far the most popular science subject that students study in Year 12. Sadly, many students fall victim to the misconception that Year 12 biology will be much like that of Year 11. However, this is quite frankly not the case, and Year 12 biology is like nothing you've ever seen before. There is just so much content in Year 12 biology that's significantly harder than Year 11. So in this video, I'm going to be covering some of the most commonly misunderstood concepts in Year 12 biology to make sure that they don't cost you marks in the end of year exam. You honestly cannot afford to let these concepts just slide under the rug and forget about them, with SACE implementing more and more application style questions to truly dissect whether you actually understand the concepts. I can guarantee that the concepts and ideas that I'm going to be covering in this video are going to be extremely important for your end of year exam, or could even be the concepts that cause you to lose marks. I know this to be the case because working with students each and every year, these are the most common ideas and concepts that cause them to lose their marks in the end of year exam. So I've got some slides for us to go through together. It really is in your best interest to put all distractions away and actually take the time to invest in learning the concepts together now, where I'm going to be guiding you through each and every one of them. If you learn the concepts now with me, it might save you a ton of time later on, or even save you from losing valuable marks in the end of your exam. It's a scary thought, but each and every year, losing just a few marks in the end of year exam is causing students to fall short from their dream ATAR or their dream grades, and you shouldn't let this be you. It's true, every mark counts. So if you don't know about me already, my name's John Kalivas and I'm the founder here at AIM Academics. I scored a 99 ATAR. Since graduating, I've completed a bachelor's degree with a double major in biochemistry and chemistry, and have since helped students just like you in Year 12 score impressive ATARs and impressive marks. I also help university students completing biology-based courses ranging from first year biology all the way to third year biochemistry. Due to my outstanding academic performance, I've managed to land myself multiple academic scholarships, including a HDR scholarship for my current master's program. I heavily attribute my success throughout my bachelor's degree, as well as Year 12, to my unique approach that I took to studying. So if you haven't watched the video where I discuss study techniques already, I would highly recommend you check out that video. This video is more so content based, where I'll be walking you through some of the most commonly misunderstood concepts from Year 12 biology. I'm going to be going through a range of concepts, but these are in no particular order. The first concept that's really poorly learned in Year 12 is CRISPR. So CRISPR was introduced to you as this fancy new gene editing tool that has revolutionized the area of genetic engineering. And this is true, CRISPR is great, but the way that students learn CRISPR in Year 12 doesn't actually do it justice. CRISPR has so many more capabilities beyond that of just cutting DNA. So hopefully you've realized in Year 12 that CRISPR can induce double-stranded breaks in DNA, utilizing its endonuclease domain. So CRISPR is actually composed of two components: the Cas9 enzyme and the guide RNA. The guide RNA serves as a template to guide the Cas9 enzyme towards a particular sequence in DNA. The guide RNA binds to a particular sequence in DNA, and that sequence contains a region known as the PAM sequence. So this PAM sequence is an important recognition sequence for correctly positioning the Cas9 enzyme at the region where it's about to cut. Once the Cas9 enzyme is correctly positioned on the target sequence, it's able to utilize its endonuclease domain to induce a double-stranded break in the DNA. Double-stranded breaks in DNA are very bad for the cell. The cell sort of freaks out and rushes to try and fix this. But when it tries to fix this double-stranded break, the repair process is very error-prone, and it's actually the cell's own repair process that introduces a series of mutations. This is where CRISPR's ability to knock out a gene comes from. It's not the fact that CRISPR induced the double-stranded break, but it's the fact that when CRISPR induced the double-stranded break, the cell tries to repair it, but it makes multiple mistakes during the repair process. So if we think about it, these could be sort of single base substitutions, insertions, deletions, all sorts of mutations. But ultimately, if we just cause a double-stranded break in DNA within the region of a gene, we can knock out that gene's function and prevent it from being expressed. This could provide some sort of therapeutic benefit if that, say, was a mutated gene to begin with and was causing some sort of disease. If we then stop its expression, we can treat the disease. However, CRISPR's capabilities extend way beyond just inducing double-stranded breaks. And these capabilities really come from the fact that we can actually disable the endonuclease cutting domain of the Cas9 enzyme. We can purposely go in there and cause a mutation in that cutting domain, inactivating it. And if we disable that cutting domain, we still have the guide RNA. So that guide RNA is still guiding the system to a specific sequence, but it's not inducing a double-stranded break anymore. And if you think about how we could take advantage of this ability for the system to be localized to a specific DNA sequence, you can imagine if we were going to fuse other proteins onto the Cas9 protein. If we fuse green fluorescent protein onto the Cas9 enzyme with a disabled endonuclease cutting domain, what we basically have is a system that fluoresces but is able to be localized to specific DNA sequences. So what this can actually enable is it can enable chromatin imaging. It can enable us to visualize where certain DNA sequences exist in the nucleus or in the genome. As well as CRISPR's ability to knock out genes by inducing double-stranded breaks, when it induces double-stranded breaks, we're also able to insert short sequences as well. This requires what we call an oligonucleotide donor, which is a short sequence of DNA that we also introduce into the cell at the same time, such that when the Cas9 enzyme induces a double-stranded break, the cell can then use that oligonucleotide donor to actually insert a small sequence into the break that we just created during the repair process. Also, you can imagine, say, if we fuse transcription factors to this Cas9 enzyme with the disabled endonuclease domain. These transcription factors could then, say, activate the expression of genes that are otherwise dormant because we've guided this entire system with the fused transcription factor to a particular DNA sequence using a guide RNA. We could also fuse epigenetic modifiers to this Cas9 enzyme with a disabled endonuclease domain. You can imagine if we fused, say, enzymes that are capable of methylating DNA. If we then guide this system with the epigenetic modifier to a certain DNA sequence, and then it methylates that DNA sequence, we could actually achieve repression of DNA expression at that region. So I hope you've developed an appreciation that CRISPR can do a lot more than just knock out genes and insert sequences. The next concept that I'm going to be talking about that students aren't really great at is the entire area of genetic engineering. And this isn't your fault, so don't feel guilty if you're not too confident with the genetic engineering stuff. This is because the content is actually quite challenging. However, there's no way that you can get away with not knowing each and every one of these concepts. So for the end of your exam, the techniques that you should be able to describe are how you can genetically engineer bacteria using a plasmid vector, how you can use a viral vector for gene therapy, such as gene editing mammals like humans or a sheep or something. You need to know about microinjection, what its purpose is and how it's performed. You also need to know how you can use the tumor faciens bacteria to genetically engineer plants in agriculture as well. I really wish I had the time to teach you each and every one of these concepts in this mini course, but unfortunately it would take way too long. However, I am going to cover just one of these concepts, which is probably one of the most important, but I will be covering all of these other concepts in our July exam preparation masterclasses. So let's explore the concept of genetically engineering bacteria using a plasmid vector. So let's suppose you want to genetically engineer E. coli, which is a type of bacteria, to express some protein Y. And let's suppose that protein Y is naturally expressed in all human cells. So your job is to propose how you would go about doing this. This is especially difficult if you have no prompts about how you would go about doing this. But if you're going to be fully prepared for the genetic engineering part of the end of year biology exam, you should be comfortable in describing this with absolutely no prompts. Okay, so let's say that we have some human cells available and we know that these human cells contain the gene encoding for protein Y. So human cells are eukaryotic cells, and hence they have a nucleus. And inside this nucleus is where you find the DNA. This is what we're interested in. This is where the gene is. So because this DNA is protected both by the nuclear membrane and the actual cell membrane, we need to actually burst this cell open or lyse the cell, and we can do this through either using what we call sonication or using special detergents. What we then end up with is a bunch of cellular debris, and amongst this cellular debris is the DNA that we're interested in. Now, the job is to isolate the DNA from the rest of the other cellular debris. And we can achieve this using filtration. So we use special filters to enrich the DNA that we're interested in, but get rid of all of the other junk that we're not interested in. So now what we've effectively got is a pure sample of human DNA, and within that human DNA, we've got our gene encoding for protein Y. So how do we then go about actually isolating just the gene encoding protein I? So what we do is we use special enzymes known as restriction enzymes. And these restriction enzymes will cut the DNA on either side of the target gene that we're interested in. So you can imagine a DNA sequence, and amongst that sequence, we've got our gene encoding for protein Y, and on either side of that target gene, we've got some restriction sites. And these are the sites that the restriction enzymes will actually recognize and induce double-stranded staggered cuts at. What I mean by a staggered cut is that it leaves these little overhangs when it cuts. And these overhangs of bases are what we call sticky ends. However, the problem is that our restriction enzymes, unfortunately, won't just cut on either side of our target gene, they also cut at any other locations in the genome where that specific restriction site sequence is present. So what we'll actually end up with is a bunch of DNA fragments where only one of those fragments actually contains the gene of interest, which is of course the gene encoding for protein Y. So how can we then isolate just that fragment containing the protein Y gene? This is where DNA or RNA probes come in. So probes are single-stranded pieces of RNA or DNA that are actually fluorescently labeled. And these single-stranded pieces of RNA or DNA will bind to your target sequence via complementary base pairing. And hence it causes the fragment that you're interested in to fluoresce. However, we're still left with lots of other DNA fragments, but only the DNA fragment that contains the gene encoding protein Y is fluorescing. So what we can now do is we can load these fragments into wells in gel electrophoresis gel. So once we load the fragments into a well in gel electrophoresis, we can then separate out these fragments by attracting the negatively charged DNA to the positive terminal of the gel. The fragments will then migrate through the gel, and they'll migrate at different rates depending on their relative size. However, only one of these fragments will fluoresce, and that's how we know that that fragment is the fragment that contains the gene encoding for protein Y. How do we then go about isolating that fragment from the gel? Well, it's actually quite straightforward because you can actually physically go in and actually cut that fragment out from the physical gel. So that then enables you to isolate your gene encoding for protein Y. So once we've got this fragment containing the gene encoding for protein Y, we would know that either end of the DNA fragment would contain sticky ends where there's a certain sequence of bases that overhangs on each side. So what we can then do is we can purchase a bacterial plasmid vector from the internet, and we can actually cut that plasmid vector using the exact same restriction enzymes that we use to isolate the human gene. So I then combine that gene with the plasmid vector that I actually cut with the same restriction enzymes that I used to isolate the target gene, and I add a enzyme called DNA ligase. And you guys would have met DNA ligase before when you're covering the DNA replication topic. And we know that DNA ligase is able to glue the sugar phosphate backbone to effectively insert this gene encoding for protein Y into the plasmid vector backbone. So once this process is done, what we produce is a recombinant plasmid vector which contains the gene encoding for protein Y. We can then introduce this recombinant plasmid into a variety of bacteria that are being cultured. In this case, it's E. coli, and we can use a technique used as electroporation to create small little transient or temporary pores in the E. coli membrane, which forces the bacteria or the E. coli to take up our recombinant plasmid vector. So now the bacteria have taken up a functional copy of the gene encoding for protein Y in a plasmid vector. So you guys should be able to represent this sort of flowchart type representation of the genetic engineering protocol for all of those other dot points that I mentioned earlier. Obviously my flowchart was very simplistic and I actually neglected a few details, but I hope it was enough to sort of get your head around the general idea. Another concept that I always see students struggle to grasp conceptually is this idea of the effect of substrate concentration on enzyme catalysis. You guys might think you're quite confident with this idea. You know, as we increase the concentration of the substrate, the enzyme activity increases up into a point which is called the enzyme saturation point, in which the rate of reaction no longer increases. But that explanation is inadequate. It doesn't actually explain what is actually happening at the enzyme saturation point. So let's think of a graph of substrate concentration versus reaction rate for an enzyme catalyzed reaction. And let's just note that reaction rates are measured by measuring the amount of product produced or reactant consumed or substrate consumed per unit time. Let's consider a super simplistic example to really illustrate the effect of substrate concentration on enzyme catalysis. So let's think about the situation where we actually have 3 enzyme molecules. You can imagine the situation where we add one single substrate molecule. This enzyme would bind to the substrate to form an enzyme substrate complex. It would then modify that substrate to produce a certain product. And let's say that that one product was produced in, say, 1 second. So this would produce a reaction rate of 0.33 products per enzyme on average. So for this catalytic cycle, one product was produced per 3 enzymes, and hence there was 0.33 products per enzyme, say per second, for example. If we then increase the substrate concentration to say 2 molecules, these 2 molecules could bind to 2 available enzyme active sites and to form 2 enzyme substrate complexes simultaneously. The enzymes will then both modify that substrate into 2 molecules of products, and hence now we would produce, say, 2 products per 3 enzymes, or let's say 0.67 products per enzyme per second. And you can see that we've now plotted that on the graph. Let's say that we then increase the substrate concentration to 3 substrate molecules. You can see that now all 3 enzyme active sites are able to bind to the 3 substrates. It's able to modify those substrates and spit them out as products. So now we produce 3 product molecules per 3 enzymes, or now it's actually 1 product per enzyme, say per second. We can once again plot this on the graph. However, what becomes interesting is when we actually add, say, 4 substrate molecules but there's only 3 enzyme active sites available instead of 4. This would actually result in one of the substrate molecules being unable to bind to an enzyme, whilst the other 3 simultaneously bind, form 3 enzyme substrate complexes, and then we spit out 3 simultaneous products for this catalytic cycle. But you can see that that extra substrate molecule had to then wait its turn for the next catalytic cycle. So by adding this extra substrate molecule, it's not actually increasing the amount of product produced per second, which is what reaction rate is. So you can see that we can actually get to a point by increasing the substrate concentration enough such that there's no longer any available enzymes to keep up with that much substrate. This is known as the enzyme saturation point. It's the point where all the available enzymes are saturated with substrate for a given unit of time, and hence we can't produce any more product per unit of time. Concept number 4, metabolic pathways, is a concept that I see students losing marks for in the end of year exam year after year because they simply cannot work out how to problem solve these problems to a good enough extent, but also their fundamental understanding as to why metabolic pathways even happen in the first place in a cell is lacking. So let's propose a very simplistic metabolic pathway where you can see A is being converted to B, B is going to C, and then C is split into D and E. And each of these specific reactions is catalyzed by its own unique enzyme. So let's consider some questions relating to this metabolic pathway. One patient was found to have a large excess of E in their body. Which enzyme wasn't working and what would they be deficient in? When we think about metabolic pathways, we can really think about it as being sort of analogous to water flowing through some pipes. If you create a blockage at one part of the water flow, the water would then compensate through flowing through other pathways. So if you think about if this individual has a large excess of E, that would mean that perhaps the water isn't flowing down the path to produce D. However, because C is still being made, it still needs to go somewhere, and we can't disrupt the smooth flux of a metabolic pathway, so it all just gets shunted into E if D is not being made. So it's likely that the non-functional enzyme is actually enzyme C, and this person is likely to be deficient in D. Another really common test and exam question is, why is a unique enzyme required for each step of the metabolic pathway? So we can ask ourselves, for example, why can't enzyme A catalyze every single reaction in this metabolic pathway? If we replace the enzyme names with some representations of the enzyme shapes, we might be able to start to think about this question. Enzymes are known to have a binding pocket or cleft known as the active site, for which the conformation of that active site is specific for a given substrate. In particular, this active site is actually complementary in its shape or conformation to a specific substrate such that they can bind together. So enzyme A, for example, would only be able to recognize the shape of substrate A and hence bind to and catalyze that first reaction. But enzyme A wouldn't be able to recognize the shape of substrate B and catalyze that reaction. Rather, a different enzyme with a unique active site shape specific for B would be required for that reaction. So this question really relates to the specificity of enzymes for their substrate, thinking about the fact that the conformation of an enzyme active site is specific and complementary for a particular substrate. Another common question that is asked is, why do metabolic pathways even happen in the first place? Why don't reactions in the body just happen in one huge, transformative step? Why isn't, say, A converted to D directly? Well, really, there's 2 reasons for this. One clear advantage of breaking up one transformation into intermediate steps is we then have the possibility for branching reactions, where one of the reaction intermediates, such as C in this case, is able to form a branch product or a byproduct like E. So if we were going to convert, say, A to D directly, we wouldn't be able to form E because we wouldn't have any C. So often in metabolic pathways, the reaction intermediates function as starting materials for branching reactions. Also, in terms of the energetics of this process, if we're going to do some huge transformation of one biomolecule to another one, that would actually be associated with a very big energy change, and either the energy requirement to do that transformation would be very big, or the amount of energy that's actually released from that transformation would be huge. So what this means is that we wouldn't be able to achieve that transformation without either putting a large amount of energy into the cell, which would overheat the cell and denature the enzymes and kill it, or without releasing a huge amount of energy into the cell as well, which would also denature all the enzymes and kill the cell. So instead, we break up the energy requirements for a transformation into small, achievable amounts at each step of the metabolic pathway, which can be achieved under physiological conditions, but also each intermediate step only releases a small amount of heat energy, which won't denature the enzymes and kill the cell. So here I've only touched on 4 concepts that are very poorly understood in stage 2 biology. I could have spoken for, say, 8, 10, 12 hours just about genetic engineering. There is a whole lot of detail that we could unpack in there, where there's so many concepts that you could be tested on in the end of your exam. However, other concepts that are quite challenging and students really struggle to grasp are things like meiosis, actually knowing how it happens, the mechanism behind meiosis, its purpose, the difference between diploid and haploid cells, problem solving when things go wrong during meiosis. Students also really struggle with the concept of the cell cycle, how it's regulated, the purpose of the cell cycle, the difference between internal and external factors that can promote the onset of different phases within the cell cycle, the positive and negative regulation of the cell cycle, and how we can progress through the various checkpoints of the cell cycle. Students also struggle with the concept of respiration and fermentation. Most students that I speak to think that fermentation is anaerobic respiration. Fermentation is an example of an anaerobic process, but respiration is something that involves what we call the electron transport chain. So it's actually technically incorrect to call fermentation anaerobic respiration. Anaerobic respiration is actually something entirely different that might occur, say, within anaerobic bacterium that live at the bottom of the ocean. However, this is only a very discrete list, and these are just a few examples that came to mind. There are many more concepts that students struggle with, and they struggle with it throughout the year in their test, but don't pay enough attention to correct their misunderstanding and then get caught out for that same very concept in the end of your exam. So I just want to reiterate that the longer you avoid mistakes or failure, the longer you will not progress. You should be addressing your mistakes and your misconceptions and the gaps in your knowledge now. You should also be addressing some of the problems with your approach to study. If the study approach that you used caused you to lose unnecessary marks in your tests, it's more than likely that you're going to lose marks for the exact same concepts in the end of your exam. So there's no better time than now during your break to refine your approach to study and take the time learning how to learn to make the process all that much easier by the time exam season comes around. So are you finding this mini course useful so far? If so, that's really good. That means I'm doing my job. But of course, my team and I can only pack a limited amount of information in this mini course. We would love to go into as much depth as we can to give you guys the greatest possible chance of achieving your dream ATAR. However, we are limited by time constraints in such a small mini course. And this is why we host July exam preparation masterclasses. We actually have 10 hours with you spread out across 5 days where we go through each and every concept covered in the year so far as well as concepts that are soon to come, highlighting the most important bits of information and highlighting also where students go wrong so you can avoid making those exact same mistakes and falling victim to them in the end of year exam. Obviously, I'm a strong advocate for these courses, otherwise I would not run them. But again, there's no obligation, and if you find enough value in this mini series, then that's perfectly fine. Also, something else that I do want to quickly mention is that we are offering free consultation calls where you can actually get into a 20-minute call with one of our team members to talk about your strategy that you're going to be taking for exams this year. This gives you a really good opportunity to see whether you're on the right track or get some advice in terms of some of the changes that you could be making to make your exam preparation better. So this is just a very chilled out 20-minute call with one of our team members, including myself, to just go over what your approach is at the moment towards your studies, especially exams, and really just a chance to connect with us and get some feedback. So there's actually limited spots for these calls due to time constraints, so I'd highly recommend you book in a time. So thanks again, guys. I hope you've enjoyed this video and learned something new, or at least it's a good refresher of some of the content you've discussed so far in the Stage 2 biology course. Thanks, everyone.